Okay, so in the last part of this chapter, part 4 of cellular respiration, we're going to focus, we're going to talk first about how other nutrients could be used in cellular respiration, and then we're going to switch focuses to anaerobic cellular respiration or fermentation. So to start off with, I just wanted to mention to you that, we use glucose, we use carbohydrates, as our molecule that we talk about because it goes through all the stages. However, I want you to realize that there are other macromolecules, other nutrients that can be metabolized and yield energy. And so if we look here in this little chart, so here's carbohydrates down the center, right? But also fats. Fats are a great source of energy. Our bodies tend to store fats, but we can break down fats for energy. And so fats will be broken down into glycerol and fatty acids. And glycerol enters at a step in glycolysis, and fatty acids enter at a later step. They enter at the formation of acetyl-CoA. Proteins also can be broken down for energy. Proteins need to first be broken down into their monomeric subunits, which are amino acids, and depending on the amino acid, they will enter at different parts of the pathway. So they might enter here, forming pyruvate. They may enter here in the formation of acetyl-CoA, or down here in the citric acid cycle. If proteins are used in this way, they will produce ammonia. So you have NH3 down here, okay, as a byproduct, which we don't see when we break down carbohydrates. So, next slide, we're going to switch gears and we're going to talk about anaerobic respiration. Okay, so fermentation. Fermentation is used in some cells in some animals and also in certain bacteria. And I left off here, certain fungi as well. Okay. So fermentation is going to be an anaerobic pathway. So when you see that term anaerobic, you're thinking does not require oxygen. And it also does not involve an electron transport chain. And so really it's only glycolysis that happens. Remember, oxygen is going to be required for pyruvate to enter the mitochondria. And so if oxygen is not present in this pathway, we don't get the last 3 steps of the respiration process. So in anaerobic respiration, we don't have an electron transport chain, and therefore we only end up producing a net yield of 2 ATP molecules per glucose. And that is going to be made by substrate level phosphorylation during essentially the first step, which is glycolysis. And so fermentation, some organisms, that's all they have to use. But it's a very inefficient process because you only partially oxidize glucose, right? There's still energy that you can't pull out of it if you're just doing this first step. Okay, so we'll look at the 2 types of fermentation. They're called lactic acid fermentation and then alcohol fermentation. And again, both of these are basically just glycolysis, and then there's a reaction, a step to regenerate NAD+. Okay, so that can be reused and recycled. So let's take a look first at lactic acid fermentation. So again, certain fungi, like yeast, as well as certain species of bacteria, will perform lactate fermentation. Okay. Okay, so here we go. So if we look over here on the right, we start off with glucose. And glucose is going to go through glycolysis. We've already covered glycolysis, so I don't want to cover it again, but just remember there's an energy investment and an energy payoff phase. And so the energy that comes out is going to be a net yield of 2 ATP. And then we also have a yield of 2 NADH molecules, just like we did in glycolysis in the first part of this chapter. Everything looks the same. We also have this end product of pyruvate, right? 2 of them per molecule of glucose. Pyruvate would enter the mitochondria and go to the second step. But in this case, we don't have oxygen, so that doesn't happen. And so what happens instead is a regeneration step. So we need to regenerate NAD+, so it's ready for the next round. And so pyruvate is going to gain electrons and protons. And we'll regenerate NAD+, and we also make, from this pyruvate, we make 2 molecules of lactate. Okay? And so the NAD+ is then ready for the next round of glycolysis. And again, this is our byproduct. Okay? We utilize certain species of bacteria. We use them to make lactate. This is used in the production of both cheese and yogurt. You may have also heard of lactic acid as it can build up in muscles. And so invertebrates like us, when we are using our muscle cells, exercising, lifting weights, whatever, and oxygen gets depleted, our muscle cells can, for a short period of time, do this. They can do lactic acid fermentation. The lactate or lactic acid, that builds up during exercise, we used to think that that's what causes the burning of muscles. Now there's evidence that's not true. And the lactic acid that's produced doesn't hurt us. Our bodies clear it from our muscle cells, and it goes to our liver, and actually our liver will pull the rest of the energy kind of out of that, use it. But yeah, so when oxygen is absent in our muscle cells, when it's been used up, our muscle cells still need ATP. And so they'll continue to do, this lactic acid fermentation until oxygen's restored. The same thing with mammalian red blood cells. So again, that includes us. Our red blood cells don't have mitochondria. And so to make energy, they'll sometimes do this lactic acid fermentation, or they will do lactic acid fermentation to generate ATP. Okay, so the second type of fermentation that we're going to quickly cover is alcohol fermentation. Okay, and so again, yeast are a great example here. Yeast are what we call facultative anaerobes, and we're going to actually, I provided you with a slide with some of these definitions on it next, but I'll go over this quickly here. So a facultative anaerobe is an organism that can switch to alcohol fermentation when deprived of oxygen. But when oxygen is present, aerobic cellular respiration is the preferred pathway, and that's because it produces more ATP. Okay? And so what happens here? So again, we start with glucose, we go through glycolysis, we make 2 ATP molecules and 2 pyruvates. So that's the same that we've been seeing. What happens next is that pyruvate gets converted into ethanol. And so you can see down here at the bottom, 2 ethyl alcohols, which is the same thing as ethanol, is produced. The first step here is going to release carbon dioxide from pyruvate. And then the second step reduces what's left, which is acetaldehyde, to ethanol. Okay, so CO2 is pulled off, and we end up with ethyl alcohol or ethanol. And that process here regenerates the NAD+. So again, this conversion of pyruvate to ethyl alcohol is just basically to regenerate the NAD+. Notice the CO2 that's produced. That's a gas. And so you can actually see that gas. That is not produced during lactic acid fermentation. So if I just flip back a slide, I'll just show you lactic acid or lactate fermenters. There is no carbon dioxide produced in this process. We do see it in alcohol fermenters, and that's actually, there's tests that are done to look for different organisms based on gas production or not. So we do have CO2 that's produced here. We use organisms that do alcohol fermentation, to benefit humans, just like we saw with lactic acid or lactate fermentation. We use organisms to help us make cheese and dairy products. Alcohol fermenters are going to be used for things like brewing and baking, wine making. Usually it's yeast, some strain of yeast that's used here. Okay, So we have two types of fermentation. We've got lactate fermentation and we've got alcohol fermentation. And again, in both pathways, it's basically glycolysis. And so again, we're getting out 2 molecules of ATP, which is better than none. So it's a way for organisms to extract energy from glucose. 2 molecules of pyruvate are always made in the process, but again, there's another step because we have to regenerate NAD+. And that step is either going to lead to the production of lactate or to the production of ethyl alcohol. If it's ethyl alcohol, we also get CO2 as a byproduct. And so I quickly just wanted to leave you with this, kind of 3 terms, and there are more terms that we could go over. If you're going to go on to take microbiology, you'll learn a longer list than just this, but I wanted to give you just a couple of terms. So some organisms are obligate aerobes, some are facultative anaerobes, and some are obligate anaerobes. And so obligate aerobes, that's organisms like us. We require oxygen for metabolism. Yeah, we have some cells that can do fermentation, but overall, most of the cells in our body do aerobic cellular respiration. Okay. Facultative anaerobes are organisms like yeast. They can switch between aerobic respiration and fermentation depending on the availability of oxygen. Again, we have some cells in our body that can do that. Short-term muscle cells can do that, but not long-term. Need more energy than they can get from fermentation. And then lastly, obligate anaerobes. These organisms are going to live and grow in the absence of molecular oxygen. And oxygen, gas to them is a poison. So if I were growing bacteria in a test tube, right, a liquid culture, I would find obligate anaerobes down here towards the bottom and obligate aerobes up here towards the top. Okay, so that's another test in micro that we use to tell if an organism is doing aerobic cellular respiration or if they're doing fermentation, where they live in the test tube, if it's a broth culture. So that is the end of chapter 7.